Good day. My name is Janne Koskinen. I'm the head of R&D at Arctia, Finland. The title of my talk is Validated Second Generation mariPOC SARS Coronavirus 2 allows rapid and automated antigen testing at least up to day 9 from the symptom onset. First, very quickly, what is mariPOC that the presentation is about? It is a fully automated immunoassay analyzer, simple to use. It consists of an analyzer, benchtop in size, and capable of random access and continuous feed analysis of samples. Very simple and intuitive user interface on a PC screen, and consumables such as test cartridges, which are inside the analyzer here. To perform a coronavirus test, it takes about 1 minute hands-on time. Nasopharyngeal swab is cut into a tube. A buffer is added from a bottled up dispenser, and the tube is vortexed to release the sample from the swab into the solution. And then a barcoded and capped sample tube is inserted into the analyzer for automated analysis. The analyzer then aspirates sample from the sample tube and dispenses aliquots of it into test cartridge, which is inside the analyzer here. And one test cartridge is sufficient for tens or even few hundreds of patient samples. The system is closed tube, so once the sample tube has been capped, there's no need to open it. And this makes it, by design, very safe for the user, even in the era of pandemic or otherwise when very hazardous patient samples have to be operated. mariPOC offers range of solutions for SARS-2 testing, such as single viral test, providing very high capacity. So 300 samples per day can be analyzed with one analyzer at the point of sampling, or then up to more than 10 pathogens, covering broad syndromic panels with flu A and B, RSVs, and obviously coronavirus. And 80 to 90% of positive samples are reported already in 20 minutes, while depending on the test, final results, negative results are reported in less than 1 hour or latest in 2 hours. And SARS-2 test has been designed to target very conserved epitopes within the nuclear protein antigen, making it very robust against strain variation. There are 3 different principles that can be used to diagnose coronavirus infections. One is the PCR exposure test, which is very sensitive but not specific for the infectious phase. Then there are antibody tests, or the so-called called immunity tests, which don't work during the acute phase, but these can be used to confirm infection later on. And then there are antigen detection tests, also called rapid disease tests. mariPOC belongs to these, and these are very specific, exactly for the contagious phase of the disease, and hence best suited for fighting the pandemic. Okay, validation data of second generation mariPOC SARS coronavirus 2 test. The manuscript has been submitted, and the pre-publication is available from these addresses. So limit of detection was verified with gamma irradiated and UV inactivated virus. The clinical sensitivity was validated in a retrospective study against qRT-PCR with specimens collected from individuals with COVID-19 suspicion in spring 2020. First cohort was 13 PCR positive samples, in more detail, dry nasopharyngeal swabs, and second cohort 45 samples were frozen UTM samples. The specificity was validated prospectively with 200 nasopharyngeal swab samples from a COVID-19 screening program. Cross-reactions were studied against microbes commonly found in the nasal cavity, and interferences were similarly studied as per the US FDA antigen template or emergency use authorization. And we were able to follow up the infection of one individual up to 14 days from the onset of symptoms. Results regarding analytical sensitivity. On the right graph, the x-axis is qRT-PCR CT value based on dilution from viral preparation with known concentration. And then on the y-axis, mariPOC fluorescence signal in arbitrary units. And as we can see, the mariPOC signal linearly dilutes in log-log graph. And the limit of detection is at around CT33 using UV inactivated virus, and using gamma inactivated virus, the TCID50 limit of detection was 2.7 per test. And the reaction volume, the test volume, is 20 microliters. Results regarding specificity and cross-reactivity. The test detected SARS coronavirus 1, which is very expected, but it showed no cross-reactivity with other microbes, and no interferences were detected. Clinical specificity of the test was 100%. Results regarding clinical sensitivity. Table 1 shows positive predictive agreement of mariPOC SARS-2 test when compared with qRT-PCR methods. So first cohort showed sensitivity of 100%, while the second cohort, larger cohort, showed 84.4% sensitivity or positive predictive agreement. And when we break down in more detail the results from cohort 2, from which we had CT values for all samples, we can see that while the overall positive predictive agreement of mariPOC was 84.4%, CT30 and lower samples, the agreement was 92% almost, and all PCR positive samples with CT less than 28, all of those were positive in the mariPOC as well. Then the pooled sensitivity from those two studies with CTs 30 or less was 94%. And if we compare that number or even the 92% at CD30 to what has been reported in the scientific literature to be the positive predictive agreement for culture against RT-PCR, we can see that this indirect data suggests that mariPOC is at least as sensitive as culture, or even more sensitive than viral culture. And indeed, viral culture could be considered as the best method to assess whether individual is contagious or not. Then serial testing and monitoring of COVID-19 infections. It's really time to challenge the dogma stating that antigen tests only work until about day 5 of the symptom onset. We were able to follow one individual with PCR and serology confirmed relatively mild infection. This was immunocompetent, middle-aged individual who did self-sampling from the nasopharynx. And we got samples from day forward. And on the y-axis, you can see the viral concentration per specimen. And as we can see, the viral concentration is high from the perspective of the highly sensitive mariPOC test until day 7, and nicely positive also until day 9 from the symptom onset, and even day 10 was detectable and would be reported as positive. Below x-axis, you can see the symptoms of this individual, and the symptomatic time and antigen test positivity correlated well. So this data shows that highly sensitive antigen tests do work well beyond day 5 and up to day 9 or 10. This was not the first time mariPOC has been used for monitoring even coronavirus infection virus levels. We published 2 years ago with our collaborators data on common coronavirus OC43 for patients having secondary infection, adults, and then 1 child with primary infection and similar results. Then some real-life examples on how this particular test has been used in serial testing. As we know, there's a global trend towards decentralized use of rapid antigen test in primary care, at workplaces and homes. Early adopters of workplace testing based on our own experience is, for example, paper industry. When they have maintenance shutdown or when they are building new facilities, new factories, they use our test to test all those incoming individuals. So for example, during a maintenance shutdown, hundreds of people from around the globe travel to this site. And of course, they pose a great health security risk for the facility. And all of those will be tested before they are let to enter the facility. And then again, also workers on site then can be tested once or twice a week to detect positive cases at the earliest and isolate them and minimize spreading of the infection. Another reference using our test for workplace testing and serial testing is global medical device manufacturer Bruker. So in conclusion, the obtained LOD was 33, which matches with the cutoff recommended by US CDC for assessing infectivity, and also approaching the theoretical analytical sensitivity of a typical PCR method with some reasonable confidence intervals, such as 95%. The highest detected CT values in clinical samples, in fact, were similar to the determined CT LOD 33. The quantitative results that mariPOC can provide give insightful data on the pathogenesis of COVID-19, and positivity up to day 10 of symptoms further validates the high sensitivity of the test. And in conclusion, mariPOC is a highly accurate test for COVID-19 diagnostics, differential diagnostics, and serial testing. And indeed, manuscripts are in pre-publication and more are in preparation. Thank you for your attention, and thank you especially for our co-authors listed here.